A warm welcome to all. So today let us discuss another very important topic related to cancer, that is genetic basis of cancer. Okay. So you all know about cancer and what are the characteristics of cancer cell, that all you studied. Okay. So in this class let us discuss what are actually the genetics behind cancer. Okay. What are the genes involved and how mutated gene leads to cancer, etc. we can discuss today in this class. Okay. So before going into that chapter, I am giving you a brief introduction regarding the cell cycle and its controlling. Okay. So you know every cell undergo the division phase. Okay. And there are resting phase also. So a cell produces by carrying out an orderly sequence of events. Okay. That is why it duplicates its contents and then divide into two. Okay. So duplicates its contents means the DNA replicates, cell organelles divides, equally distributed, and a cell divides into two. Okay. So that are all events taking place first. Then this cycle of division, duplication and everything, that's known as cell cycle. Okay. So cell cycle is an essential mechanism for every living cells to reproduce. So in the somatic cells as well as in the germline cells. Okay. Now most of the time a cell cycle spend in the most time is in the interphase stage. Okay. And interphase, you studied in your UG classes, divide into G1, S and G2 phase. Okay. G1 and G2 are the gap and S is the synthetic phase. Okay. So in the S phase, that is very important one, synthetic phase, and others two are known as gap phases. Okay. Now you may be heard about G0. What is G0? So it is a resting phase, or you can call it as quiescent stage. Okay. So it is not dividing here, not preparing to divide, etc. When a cell is not dividing, it enter into the G0 stage. Okay. So until getting any signal. Okay. So when a cell get a signal to divide, then only it passes to the next stage. Otherwise, it will be in the quiescent stage or inactive stage, that is the G0 cells. Okay. So we have to think how this is happening. There are a particular signaling system when only a cell passes into the division stage. Okay. Otherwise, it will be in the sleeping mode. Okay. So this is when requirement is there, then only it passes to the division phase. So that is a controlling system. Okay. So to ensure that they replicate all their DNA and all their organelles and divide in an orderly manner, every eukaryotic cell possesses a complex network of regulatory proteins that are known as the cell cycle control system. So this network of system or these regulatory proteins, they monitor if the cell division is perfect. How? In order to have a perfect cell division, there need clear DNA replication, cell organelle division, everything. If all these are in perfect condition, then only a cell starts to divide. Okay. So this is under the control of particular group of proteins, and they are known as the cell cycle control system. Okay. So to ensure, it ensures the events of cell cycle, that is DNA replication, mitosis, etc. So a set of sequences, and each process is completed before the next one begins. So what are the events that should happen in the G1 phase? That should be completely or that is full finished, then only it passes to S. Then what are the things happening in this? Everything finished, then passes to G2. There are certain controlling systems that will control this. If finished, confirmed, then only pass to the next. Clear? So this is the cell cycle controlling system. Now there are certain checkpoints. So in between these different phases, and these checkpoints are the surveillance mechanism that halt the progress of cell cycle. Okay. So if they are happening, any of the chromosomal DNA is damaged, or any that is by mutation or what, something happened. So replication is incomplete, or any damage detected, or a certain DNA replication S phase or chromosome alignment in the M phase is not proper. What happens is these checkpoints they control. So you know, when we are traveling for one state to another state, there are certain checkpoints or checking areas. So what they are actually checking, enough our license, our papers, everything is perfect, then only they will allow them to outside. Okay, and nowadays there are COVID protocol, if you are vaccinated only you will be allowed. Likewise, there are certain checkpoints that will determine whether this is able to pass from this phase to next phase. Clear? So these are very, very important, these checkpoints. So any abnormality, any mistakes in these checkpoints will lead to that problem to proliferation of cells, uncontrolled cell division, etc. Okay. So likewise in our cases, human beings as well as other organisms, this is happening. Clear? Okay. Now about the checkpoints. So here we want to discuss more about the checkpoints. So there are checkpoints between G1 to S phase, G2 to M phase, and the M2 during the division stage. So there are mainly 3 checkpoints important, and these checkpoints are the control systems. So that confirms that environment is favorable for proliferation before committing to DNA replication. That is G1 to S. In the S phase, replication is going on. So is that cell is enough, or that can be passed to the S phase. If it is completely alright, then only it allows. That is G1 to S phase gate or checkpoint. Then another one is in the G2 to M phase. So this control system confirms that the DNA is undamaged. That is DNA already replicated in the S phase, and after that here it is checking that DNA is undamaged, fully replicated, and it is perfectly alright to pass to the division phase. Okay. Then during mitosis, in that stage also, there is a machinery that will check up if they are the cytoskeleton machines, spindle fibers are perfect, or they are aligned in the correct position, chromosomes are in condensed stage, and everything is checking in this point. So after checking only it will allow, you can go now, okay, to the next stage. So these are very, very important. So major checkpoints are there. Now, so entry of a cell into M phase is initiated. So all these completed, and now the entry of a cell into the division is initiated only when there are certain proteins called maturation promoting factor, that is MPF. So this protein should initiate this process. You studied there are promoters that will bind, then transcription occur, translation occur, and protein produced. So they will get a signal to produce the protein. So that is here is an MPF. Then only it will start. Okay. So MPF is an important protein, that is maturation promoting protein. It has 2 subunits, and that subunit is one with the kinase activity and another with the cyclin. Okay. So cyclin and the kinase are very important because cyclin having no enzymatic activity. So this unit, this will function only when they work together. Okay. So that is the complex of forming the cyclin and kinase, and the kinases of cell cycle control system is therefore known as cyclin dependent protein kinases. They are not cyclin dependent protein kinases or CDKs. Cyclin dependent protein kinases or CDKs, very important one. This CDKs is actually initiates or they will give signal. So CDKs or cyclin dependent protein kinases are important one in the cell cycle controlling system. Okay. So what are the various CDKs and associated cyclins? So in the first checkpoint you can see there are CDK4 and CDK6 with the cyclin D. So CDK4, CDK6 either 4 or 6 combined with the cyclin D. So that is the first checkpoint. Okay. In the G1S transition, in that stage you can see CDK2 combined with the cyclin E, CDK2 combined with the cyclin A in the second check phase. And there are 2 cyclins. And in the third checkpoint you can see cyclin A CDK1, cyclin B1 CDK1 complexes. So in all these stages, these complexes controls, this protein system controls whether it is able to pass to next. They will give the signal. Okay, they will produce in that time. Okay, so these are the initial, introductory part regarding this. So you can see in this image what are the different checkpoints. See, G1, S, G2 and M. Here one checkpoint, here one, here one. Okay, so this is a G0 phase. And you can see here different types of cyclins and CDKs in each stage, and these are all different types of proteins and activated genes. And the next stage, how much time they are taking in each stage. Okay, so CDK2 and A in this checkpoint, see from here to here. This is the checkpoint from here to here. Okay, likewise every part having a checkpoint and the time also. Clear? Okay. Now, so up to this is generally this, up to this I am given this portion for controlling what are the systems that control cell cycle, because you know any alterations in the cell cycle. So whenever cell want to divide, or is there any requirement of cells, more number of cells, then only it want to divide. That is under the control of the first signaling systems or cell cycle factors. Okay. Now look, in the case of cancer you studied, it is an abnormal division, abnormal differentiation, abnormal proliferation, metastasis, everything you studied. So here actually what happens is there is no controlling system, or checkpoints are having some problems. They are in abnormal behavior. So that's why I discussed the checkpoints in the first part. So something not happening properly. That's why there is an abnormal division. So what are the reasons behind that? Okay. So let us study the genetics of cancer. So the genes that induce cancers are generally we call them as oncogenes. Okay. So there are two groups of genes that are cause carcinogenesis. They are oncogenes. You can call them as cell accelerators. You know what is the function of accelerator? When we drive a vehicle, so when you press or you give stress to accelerator, how much intensity, that much speed your vehicle will pass. Okay, so that's an accelerator. Then tumor suppressor gene, they are the brake. Brake means you will control it. See, your speed you can control by pushing the brake. See, so there are accelerator as well as brakes. That are the two groups of genes. So that oncogenes and tumor suppressor genes. In the name itself you can see tumor suppressor genes, what function actually they do. They will suppress or they will reduce the formation of tumors. Okay, so they will repair the mutated gene, or they will kill the mutated gene, or whatever the process. Okay, so they will check out it if any problem or anomaly. That is tumor suppressor gene. But what about oncogenes? They will accelerate the cell division or cell growth. These are the two categories. Okay. So in every cell, there are certain genes. They are the proto-oncogenes. They are actually there in the sleeping mode or somewhere else. But when a process like mutation causes, these proto-oncogenes are able to convert into oncogenes. That may lead to cancer. So how these proto-oncogenes able to become oncogenes? That's our question. Okay, how this is happening? There are various types of mutations happening. A sudden change. Sudden change may be by radiation. We already discussed the causes of cancer. Whatever it may. Yeah? It can induce change in the proto-oncogenes and convert them into oncogenes. So the mutation may be point mutation. What is point mutation? In a particular focusing, a particular gene only lost. Okay. Or it may be chromosomal rearrangement. That's by translocation from one chromosome to another. Or two genes in the same chromosome. Or maybe duplicated or amplification. Or maybe epigenetic modification. Whatever it may. So many types of mutations, aberrations may be happening. This can lead to oncogenes. Clear? So this is the basic thing. Now look. So in detail. So oncogenes, it may be by deletion, translocation or point mutation, and maybe an abnormal gene product. by combination of any two genes resulting into an abnormal gene product. One example is Philadelphia that we will discuss, and myelogenous leukemia. They are the examples. Then actively or more active by gene amplification. Okay, normal product is overexpressed. That's one example is NMYC amplification, 700 times increase in neuroblastoma, a type of cancer. Okay, so genes, basically genes that promote autonomous cell growth in cancer cells are called oncogenes. So their normal cellular counterparts or proto-oncogenes are already there. They undergo changes and become oncogenic, and cell results in more proliferated condition or abnormal cell division. Okay, so they are characterized by ability to promote cell growth in the absence of normal mitogenic signals. Okay, so there are already signals that we discussed that may be harmed or whatever it may be. So that may result into an abnormal division. Okay. Now, so these oncoproteins, they resemble normal products of proto-oncogenes, but they have no regulatory elements. They have no regulatory elements. That's a difference. Okay. So their production in the transformed cells become constitutive. They not depend on growth factors or any external signals. So no external signals can control them. Clear? Okay. So proto-oncogenes can be converted into oncogenes by several mechanisms, including mutation as well as amplification. Maybe deletion, duplication or translocation, or maybe gene amplification. Clear? See, in this picture you can see a proto-oncogene by normal cell growth. On one side you can see normal cell growth, another proto-oncogene become mutated, and what happens is normal controlling system, or they are out of the normal signal system and they lose that normal condition, and they that growth control is not there. It will grow abnormally and results in cancer. Clear? Okay. Now, what are the basic means in detail regarding these genes? There are, I already told you, two conditions. One is activation of oncogene. Second one is inactivation of tumor suppressor gene. Okay. So what is tumor suppressor gene? I already told you. They are the genes that control the abnormal growth or that repair the mutated DNA. But what happens is, when they are active, then only they can control or they can cure or they can treat or they can rectify the changes in the DNA. Okay, and the cells become secured. But what happens when these tumor suppressor gene inactivated? No, it will lead to cancer. Abnormal cell division means mutated gene will pass to, mutated DNA will pass to next stage. It will continue and cancer will be the result. Okay, and activation of oncogene is another condition. So there are two types of oncogenes. Mainly we will discuss. One is RAS gene, RAS gene, another one is MYC gene or MYC gene. And inactivation of tumor suppressor gene, there are two tumor suppressor gene, not only two, three are there, P53, APC and RB, that is retinoblastoma. So we will discuss P53 and retinoblastoma. Okay. So first of all we can discuss activation. So activation of that RAS and the MYC gene. So what is actually that? It allows the cell to bypass the checkpoints. I already told you what are checkpoints. Okay. So when these genes are activated, automatically without any signal it passes to the next stage. For example, G1 to S or S to G2. Okay. So allow cell to bypass when these genes are mutated. Clear? So actually what is RAS is, they are the GTPases, guanosine triphosphate, GTPases, and they are the molecular switches in cell. They regulate the signaling pathway. So GTPases are molecular switches, they regulate the signaling pathway. Okay. So mutations causes these genes to turn on, and there is an overproduction of the protein. So RAS gene that will turn on, and there is an overproduction of certain proteins it increases GTP binding and leads to overactivity. So overactivity means what? That you are ready now, you can go to next stage. So they are giving signal continuously. So cell starts to continuously divide, proliferating, undifferentiated clones are forming. That's actually what cancer. Okay. So growth factor stimulates RAS protein that will produce more, transcription occurs, CDK cycling complexes and overproduction. This is happening when this RAS is more activated. Clear? More about RAS. So RAS is switched on by incoming signals. It switches on, turn on genes involved in cell differentiation, growth and survival. Okay, mutation in RAS leads to production of permanently activated RAS. RAS are in two forms. One is in the activated, second one is deactivated. That is GDPase and GTPase. So in the GTP condition they are activated, but in other they are inactivated. This process is continuously changing. When required only activated. But what happens is when mutated, it's permanently become activated. So it will switch on and it will it will give proteins and give signals for uncontrolled division. Okay, so there are 3 RAS genes usually located in chromosome 17. Three of them are well studied, that is HRAS, KRAS, NRAS, based on the name of the scientist who discovered first. That is the first alphabet. Okay, so mutation that permanently activate RAS are found in about 20 to 25% of human tumors and 90% in certain type of cancer, especially in the pancreatic cancer. Okay, so it activates several pathways like kinase activities and its transcription of genes that results in the proteins that will enhance cell growth and division. In the off state, it is in GDP condition, and the off state, it is bound to GTPase. So GTPase means phosphorylated condition. Okay, GDP to GTP, phosphorylated condition, extra phosphate group. Okay, so there's products involved in kinase signaling. That's important one. To turn on means it bind to GTP. Okay, to turn off means it bind to GDP. See the image, I will show you the image. See, RAS normally turn on means bind to GTP. That is inactive RAS GDP. But RAS to GTP is what? Bind to GTP is active. In that condition, controlled growth, proliferation and migration. This is the normal case. Suddenly it become GDP when it got signal. Okay. But what happens is here it is in mutated condition. It permanently bind to GTP. It become active. This reverse process is very less or absent. Only this process, that is GTP binding process. So uncontrolled growth and proliferation that may leads to cancer. Clear? Okay, I hope you understood well. Now, so these changes are stuck in on position. That's very important. On signal. You have to remember RAS in on, activated. GTPase complex. So cell growth and proliferation. So expression and amplification leads to continuous cell proliferation. So RAS transcription increases, excess gene protein and positive signal for cell division. So RAS transcription increased, protein signal for proliferation. You continue, divide. So ultimately what happens is more cells, uncontrolled growth result in the cancer. Clear? So now, so that's all about RAS gene. Now second one, I already told you about a tumor suppressor gene. So one very important one is P53. So what are tumor suppressor genes? So in a cell, actually there are two conditions. One, when a checkpoint detects that DNA after replication reaches to S phase, before going to M phase, the checkpoint determines there are some mistakes in the DNA. There are some mutated parts or some mistakes. So the cell will think how we can rectify this. So there are different options are there. So think about damaged DNA or whatever it may. In some problems, it's not in a good condition to pass on to the division. So in that stage, what they will do? There are different options in front of the checkpoints. One is they try to repair it. That is DNA repair. You studied DNA repairing mechanisms. Okay. So they will think about it and they will start working for that, and they will find out our energy will waste and no scope for repairing. So what will they do is, next step is the programmed cell death or apoptosis, means the cell have to die, otherwise it will cause problem to the new forming daughter cells, etc. So they will decide to kill that. That is programmed cell death or apoptosis. Next. Okay. So these are the two cases in first one thing. And another one is, if this is not possible, that what is the third option is, it will produces a transcription protein, and the transcription protein will inhibit CDK cycling, that complex. You studied that. That will control the cell cycle signaling system. So it will inhibit that protein, that function, and cell cycle inhibited. Clear? So this is the way this tumor suppressor gene generally act and prevent the formation of abnormal cell division. Okay. So this is the normal case happening in a cell. See, so how beautifully it controls. You just imagine. So this is the way most of the people, human beings and other organisms, they are well protected. That is there in our body itself, tumor suppressor gene. Clear? So I hope you got a clear idea regarding that. It's already there. Okay. Now think about the mutated condition of this tumor suppressor gene or P53. When it is mutated, what happens is, this gene, it become inactivated. This function will become inactivated. So no protein formation, no transcription, no protein. It will not code and not produce protein, no promoter, etc. So that will lead to that mutated, the DNA will pass to the next stage, replication. After replication, it will pass to mitotic division. Division take place and leading to cancer. This is tumor suppressor gene inactivation. See, so this is a second case. Clear? Okay. So about TP53, that's very important one, tumor suppressor gene that produces the protein P53 protein, and PRB is another one, another tumor suppressor gene that produces retinoblastoma protein. So you have to study two important one. One is P53, another one is PRB. So retinoblastoma. So this is first that cancer, the name retinoblastoma, from that it is detected. Okay. So if any proto-oncogene mutated to oncogene, this will act. Okay. So when a proto-oncogene become oncogenes, this gene will generally act. Okay. So they will try to suppress it. So there are DNA damages. Response proteins give signal to P53 to activate. It will take action. What are the actions? Repair mechanism, apoptosis, or arrest cell cycle. Anything from this, anything they will choose and rectify it. This is happening. That is about the tumor suppressor genes. Okay. Now about P53, that's very important. P53 protein that binds to DNA. So P53 protein is produced by the TP gene that produces this protein. When it produces, if any mistakes found, then that stimulates the gene to produce a protein. That protein is known as P21. That interacts with the cell division stimulating protein CDK2. Okay, so it will bind with the DNA. It acts as a promoter and bind with the DNA, and transcription take place. It will produce a protein, that's P21. P21 interact with that CDK2 and control the cell division. Clear? Okay. So another peculiarity is it is an unstable protein. It produce degrade continuously. Okay. Then when P21 is complexed with the CDK2, cell cannot pass through the next stage of cell division. Okay. What is CDK? You know that, cyclin dependent kinases. That's its function, already we discussed in the first half. So P53 is an unstable one. So when it produces any problem detected, it binds with the promoter and produces a protein P21. P21 bind with the CDK, and CDK will control it. When it complexes with the CDK, it will never produce or it will never pass to the next stage. Okay. So if everything found correct, there is no need of formation of P21. Okay. If the cell is in perfect stage, after S phase, DNA is in normal condition, no need to produce what? No need or no formation of P21 required. Clear? So, mutant P53 can no longer bind DNA in an effective way. Okay. The cell divide uncontrollably and form tumor when there is a mutant P53. Okay. So, when P53 become mutated, what happens is it never bind, it never act as a promoter, never bind DNA and never produce P21 and that no signals. So, what happens? Cell divides and result in tumor. Okay. The last one is retinoblastoma protein. This is also a tumor suppressor gene producing protein. So, this is occurring in the human beings also. There are RB1 and RB mainly tumor suppressor protein. There are one function is to prevent excessive cell growth by inhibiting cell cycle. Same way as the last case. Okay. And when cell is ready to divide, what happens is it is phosphorylated. When cell is ready to divide, it becomes phosphorylated, inactivating and cell cycle allowed to progress. Okay. When cell is ready to divide. If the cell is not ready, some problem detected, what happens is it becomes activated. Activated and cell cycle arrested. Same way what in the last case. Then this makes a protein that is PRB. RB gene produces a protein PRB. RB is retinoblastoma. It helps in stopping cell growth, growth. Okay. So, each cell normally have 2 RB1 genes and usually if both RB1 genes are mutated, it results in the disease, this cancer retinoblastoma. Okay. When both of RB1 mutated, missing, it can grow unchecked, result in cancer. Okay. In humans, this RB is located in chromosome 13. Okay. So, when both alleles of the genes are mutated, what happens is this RB, that protein not producing and promoter binding not taking place. What happens is the cell division continuously going on, result in cancer, mainly retinoblastoma. Okay. That is all about tumor suppressor gene. So, tumor suppressor gene P53 and RB, then RAS gene, that is the activation gene. So, you have to study both and first part the cell cycle signaling. Okay. So, ability of PRB to bind important protein, that is E2F, E2F dependent transcription. So, PRB is binding E2F protein, that is a promoter. Okay. So, it inhibits cell cycle when required. Okay. And PRB restricts the cell's ability to replicate DNA. That is important one. Then this happening in the G1 phase itself, first gap phase to S phase, that is synthetic phase, this will happen. Okay. So, PRB binds and inhibits E2 promoter binding protein. That is the thing happening here, and it binds and transcription factor of E2F push the cell into S phase. So, if it is normal, it will push into the next phase, otherwise it will stop. It will give the signal to stop the cell division. So, these are about a tumor suppressor gene. Its inactivation and activation of RAS gene are very important two things regarding that genetics of cancer. Okay. So, that's all for today's class. So, have a nice day.